Dr. Evans is a James McGill Professor of Neurology and Neurosurgery at McGill University and a researcher at the McConnell Brain Imaging Center. He's a co-director of the Ludmer Center for Neuroinformatics and Mental Health, attending to the neuroinformatics components with the 60+ strong team at the McGill Center for Integrative Neuroscience. Here he will briefly introduce some aspects of the role of his team in the BigBrain project.
Good morning, everybody. It's a pleasure to have you here at the second BigBrain Workshop. I do apologize for the weather outside, but consider yourself lucky we haven't got 6 feet of snow out there, so. Okay?
Yes.
We're good. All right. Thanks very much, Natasha. So I'm going to give you just a brief introduction to the history of the big brain. I'm not going to say too much about what we're doing right now because a lot of that will be presented by various people through the course of the day. It is a pleasure to. So the big brain, the origins of the big brain. We started at the MNI 30 years ago, developing atlases with MRI of various forms. And if you want to read more about brain atlases and templates, you can go to this review article. The problem is that they're they're with MRI. So they're three-dimensional, which is great, but they're at 1 millimeter resolution. The other end of the universe, we have atlases of small portions of brain, which are based on histology, myeloarchitectural maps of small parts of brain, either slices or three-dimensional blocks in a small piece of brain. So neither of those are ideal. We would like to have a three-dimensional histology map at 1 micron resolution. And that's at this moment beyond our reach, not too far, but it's beyond our reach at the moment. The compromise that has been arrived at so far is the big brain. And the big brain was published in 2013 in Science. This was a collaboration between the Jülich Research Center, Katrin Amundsen and her team, and my team here, with the continued involvement of Professor Karl Zilles and his team. And you'll be hearing more from Karl shortly. So the big brain represents a compromise between those two extremes of histology and in vivo 3D imaging. Katrin's team at the Jülich Research Center did the wet work, if you like. They did the sectioning in a single post-mortem 65-year-old brain into 7,404 sections. The data was digitized and then sent to my lab. And this started at this point, almost 10 years ago. And my lab, particularly Claude Lepage and Lindsay Lewis, did the work of integrating all of those 7,000 sections into a coherent three-dimensional whole. The data set has been downloaded at this point over 25,000 times. So we placed it in the public domain and it has become a global standard. The good news is that lots of people are using it. For us, the bad news is that we don't know what they're doing with it, which is why we're having a workshop here to find out what you're all up to. I think that's a good example of the pros and cons of open science. This is a video. Perhaps it's not working. No, it's not working. Not to worry. This is the kinds of individual slice data that we were faced with at the outset of this project. Almost all of the slices had some problem or another, whether it's gross rips and tears or geometric distortions or optical imbalance. And that's put a lot of work, a lot of effort to realign, reconstruct, and establish the optical balancing. Again, you'll hear more about this from various people in the presentations. So the net result was this. And again, to make the point, on the left, you have a comparison of an MRI volume at 1 millimeter. And in the bottom left, you can see zoom in to that image. And of course, that's at 2 millimeters across that box. And each of one of those pixels is 0.5 of a millimeter. So that's already higher MRI resolution than we're used to seeing. On the right, you see the same zoom for the big brain. And of course, you can see almost cellular resolution on the right. So that gives you an idea of how we can go from whole brain to individual almost cellular resolution. If you extract the surfaces, this is what you look, and this is what you obtain. This on the left-hand side, you see the real surface, and on the right, you see the inner and outer cortical mantles, surfaces of the cortical mantle. With Louis Bourgeon and his team at the National Research Center in Ottawa, we developed the Atelier 3D visualization tool, which allows us to explore this data set. Bear in mind that this data set is the equivalent of 125,000 MRI volumes. For those of you who wonder about that, this is a 20 micron volumetric data set. If you compare that to MRI imaging at 1 millimeter volumetric, that's 1,000 microns in each dimension. You go down by 50, a factor of 50 in each dimension. So 50 times 50 times 50 is 125,000. So this data set is the equivalent in size to approximately 125 ADNIs, for those of you who know what the ADNI database is. We place the data set onto the web for download using the Loris tool and this interface. And as I said, it's been downloaded so far by more than 25,000 groups. We've had 2 releases. The original release in 2013 after the Science paper was followed up in 2015 with release 2.0, where we improved upon a number of issues related to linear registration and the optical imbalance. And this data was also aligned in MNI space and made available to the community. We've provided it in different levels from 100, 200, 300, and 400 micron resolution in native and MNI space coordinates with tissue classification information, cortical surfaces, and documentation to describe what's been provided. A brief comparison, this is the 1.0, and you see the optical striations, and 2.0, those have essentially been removed. This again is the Atelier 3D visualization tool, and here this shows us we can provide oblique slice planing through this volume. And for many people in the audience are particularly interested in the hippocampus, so you can see that we're aligning this oblique slice along the long axis of the hippocampus. And if I zoom in, that's again along the long axis of the hippocampus. Well, I guess you hear more from many people about the hippocampus, Roseanne, right? There's now a proliferation of visualization tools which are being used to examine the big brain. Atelier 3D that I've talked about, Neuroglancer, which you're familiar with, and the BOSS is a spatial database in the cloud that serves big brain. And so now people can explore this 1 terabyte approximately database through the web using the BOSS. And Greg Kiar is in the room. Greg and his former team in Johns Hopkins University developed the BOSS, and we're happy to be able to use these kinds of technologies. The last thing we want to do is to reinvent the wheel. And so far, this is still in prototype, but it could be a lot of fun. Armin? Where's Armin? Armin is one of the young coders in the lab, and Armin has been working with the LEAP technology. You can see the device here. It's not very expensive, but this allows you, to capture hand motion and hand signals to define particular instructions to the software so that you can rotate or zoom simply by hand motion. So we're basically creating a minority report interface to explore the big brain data set. I'm not sure ultimately what will be the scientific advance that comes from that, but it's a lot of fun, and it means that the donors are very happy. So to close, what are the potential applications of the big brain? There are many and varied, but the automated 3D cytoarchitectural analysis is possible. Neuroanatomy teaching. It's a stereotaxic template, obviously downsampled and segmented. It's a gold standard for image processing algorithms. It's a master data space for combining information across major projects such as the Human Brain Project in Europe. And it's a prior for neural modeling. You'll hear examples in all of these areas in today's talks. Of course, I can't end without acknowledging all the people in my lab who do all the work that's being done here, particularly Lindsay, who's staring out of here. And I don't know where Claude is. Yeah. Claude, stand up, Claude. I think everybody should know that Claude spent years doing this. So that's all I want to say by way of introduction. It's now my pleasure to introduce the keynote speaker for this workshop, Professor Karl Zilles. Karl has won so many prizes, you can't count them all. He is, in my view, the world's greatest neuroanatomist. He hates it when I say that. What I find fascinating about Karl is that he's a classically trained German neuroanatomist. Everything that implies, meticulous attention to detail, encyclopedic knowledge of the history of neuroanatomy, and a constant source of information for all of us who work with him. But at the same time, as being a classically trained neuroanatomist, Karl has embraced the newest of new technologies in terms of multi-domain characterization of brain chemoarchitectural maps, brain fingerprinting with various receptor and transmitter maps. And he speaks quite happily about dendrograms and partitioning. I don't know many neuroanatomists who use this kind of language, and I think that is a testament to his creativity and his openness to new ideas. It's been a pleasure working with Karl for many years. He's now, in theory, the senior scientist at the Institute for Neuroscience and Medicine at the Jülich Research Center and at Aachen University. And in theory, that means that he is now being booted upstairs to be an emeritus professor. In fact, all it has done is released him from administrative duties to cause trouble everywhere around the world with his ideas and his questioning. And it's always a delight to listen to Karl give us the latest on his thinking about brain. So, Karl, over to you.
Okay. Thank you, Alan, for the introduction. It makes me always feeling strange when you speak about my person. So I would like to show you what we have done during the last 10, 15 years to go a new way of understanding the cerebral cortex. I was in the beginning, together with Catherine, involved in the development of cytoarchitectonic methods, and it was a pleasure to do it over several decades and to develop new techniques, new methods. But then I thought, well, we need some more information about the cerebral cortex, some information which is nearer to function, to understand the combined structural functional organization. And so the idea came up to postulate and to study the organization of the cerebral cortex by looking at transmitter receptors, not transmitters on the looking to the receptors for transmitters. The reason is quite simple. Transmitters are or have a considerable amount of ambiguity regarding their function. The function completely depends on the receptors on which the transmitter is binding. So a transmitter can be inhibitory and excitatory depending on the receptor type on which the transmitter binds. And so we thought that receptors may be key molecules of signal processing in the brain, and we should know whether the receptors are homogeneously distributed throughout the brain or they show heterogeneous distribution, whether this distribution is correlated with functional subdivisions of the brain or structural subdivisions. So the aim was to combine all what we have done with cytoarchitecture with receptor architecture, and that's what I would like to show you today. What can the receptors tell us about the human cerebral cortex, its organization and function? Let's start with a first view into this relation, and I have selected the amygdala. What you see here is a 4 Tesla image of a post-mortem human post-mortem brain, and you can easily see the amygdala up here. And you can see that the amygdala is subdivided, even in the structural images, into a denser medial part, into a less dense intermediate part, into a intermediately dense lateral part. And now let's focus on the amygdala on this side. In immediately following sections, alternating sections, we have reacted the tissue for receptors. And the receptor density can be absolutely quantified by using the method of in vitro receptor autoradiography. That's a radioactive technique where specific ligands are labeled with tritium, and we can translate the intensity of radioactivity in receptor concentrations in femtomole per milligram protein. We can color code these densities. So what you see next is this part of the human amygdala, and first you will see the muscarinic M2 receptor. That's an acetylcholine receptor, and you can see that the lateral part of the amygdala has a very low density. We followed the spectral sequence of colors, so blue is always low density. Then you see the intermediate part here, which has a very high density, and the medial part which has a mixture between low, intermediate, and very high densities. So this image tells us immediately that the amygdala is not a single structure. The amygdala is a complex of different structures with completely different receptor expression, at least for the cholinergic muscarinic M2 receptor. And you see that this is an immediately following section by focusing, for instance, here on this part of the cortex, which appears here and does not belong to the amygdala. That's the amygdala. Now the question was, are all receptor types distributed in the same way in the amygdala? with an intermediate density in the basomedial part, very high density in the basolateral part, and a very low density in the lateral part. And the answer is no. If you look for the inhibitory GABA A receptor, which is the most important inhibitory molecular mechanism in the cortex, you see that the lateral part has the relatively highest density, the intermediate part the lowest, and the basomedial part and mixed intermediate density. Then we go to the next receptor, which is an excitatory receptor, the glutamate NMDA receptor. And you see again that we have very different distributions, and even the lateral part of the amygdala can be further subdivided by its concentration of the NMDA receptor subtype. That this has to do something with connectivity can be shown here. In the upper left, you have an image from Weismann and Lichtman about the structure of the human hippocampus with the CA1, CA2, CA3 regions, and the dentate gyrus with the molecular layer called MOL here in this image. The hippocampus receives its input, its only input, from the entorhinal cortex. So all information in the neocortex is converging on the entorhinal cortex, and from here a fiber tract called the perforant path goes into the hippocampus. And this perforant path PP in the upper left image ends mainly in the molecular layer of the dentate gyrus, at least in the human brain, but also in the CA1, CA2 region of the hippocampus. And there, after synaptic switching, the next fiber tract is the fiber tract called Mossy Fibers. The Mossy Fibers originate in the dentate gyrus, and they end in the CA3 region, and only in the CA3 region of the hippocampus. And the last circuit starts in the CA3 region, and via Schaffer collaterals of the axons of the CA3 pyramidal cells, this fiber tract ends in CA1. Now let's go to the receptors, and I would like to focus your interest first on the lower right image. That's the kainate receptor. The kainate receptor is a glutamate receptor. And what you see is that nearly selectively, the CA3 region of the hippocampus is labeled, where the synapses of the Mossy Fibers are found. Also, there is a high labeling in the hilus of the dentate gyrus. Also there, the Mossy Fibers are ending. So the kainate receptor more or less selectively labels the second step of the intrahippocampal circuitry from the molecular layer of the dentate gyrus via the Mossy Fibers to the CA3 region. Now let's go to the Ampa receptor. The Ampa receptor is the leading excitatory receptor, glutamate receptor, and you see a strong labeling of CA1, CA2, CA3, and the molecular layer. So this receptor labels all three synaptic stations of the intrahippocampal circuitry. And then finally we go to the NMDA receptor, a receptor very important for studying learning mechanisms. And the NMDA receptor does not label with highest intensity the CA3 region, where the kainate receptor has been found, but the CA1 region and the molecular region. That means the first and the last stations of this circuitry. So this was the first finding we did, where we got hints that receptors may label functional circuits, microcircuits, or larger functional systems. Now, the next question we worked on is, does the receptors subdivide the cerebral cortex into major compartments? For instance, into primary sensory cortices. These are cortical parts of the neocortex where the thalamocortical, the very functional, very specific thalamocortical fibers end. So, for instance, the optic radiation from the lateral geniculate ends mainly in the primary visual cortex. The acoustic radiation from the medial geniculate body ends mainly in the primary auditory cortex. And the thalamocortical fibers from the somatosensory thalamic nuclei end mainly in the primary somatosensory cortex. So is this concept of the cortex, of primary areas and of secondary or higher multimodal association areas, reflected in some way by the receptors? And here you see an image on the left side of a coronal section through the human cortex, and on the right side also, but the left image is from the central region with the central sulcus. Central sulcus can be seen here. And the left image is from the occipital region with the calcarine sulcus here. And you see immediately that the muscarinic M2 receptor is very densely expressed in the primary visual cortex around the calcarine sulcus, and it sharply delineates the primary visual cortex from secondary and other visual cortices. And when we go to the left side, here's the central sulcus. This is the anterior wall of the central sulcus. This is the posterior wall. You see a very high density in the posterior wall of the central sulcus, and this is the primary somatosensory cortex, sharply delineated. And here we have the primary auditory cortex. Where is my, here. The primary auditory cortex on the Heschel gyrus, which is cross-sectioned in frontal section and looks like a mushroom. And here in this Heschel gyrus, you see again a very high density, absolute density of the muscarinic M2 receptor, more than 400 femtomoles per milligram protein. You see also that the motor cortex, which is here, has a very low density, and other cortices have densities which are in different cortical layers, or they show different layer patterning like the entorhinal cortex and so on. Now we put the question forward to see what other receptor types are doing in a primary sensory cortex, and the example is the human primary visual cortex. Here you see again, at higher magnification, the distribution of the muscarinic M2 receptor as shown, as I have shown you before, and you see that this receptor sharply stops at the border between V1 and V2, here at the upper border, and the other arrow shows the lower border between V1 here and V2. You can also see that the receptor is not equally distributed throughout the cortex, but seems to have a layer patterning. Then we look to the noradrenergic alpha-2 receptor, that's shown here, in an immediately following section through the same human brain. And you see again that even the alpha-2 receptor delineates quite sharply the primary somatosensory cortex from the primary visual cortex, sorry, from the secondary visual cortex. But here, the high density of receptors is restricted to layer 4C. Layer 4C is the layer where most of the geniculocortical fibers are terminating. And even the inhibitory GABA-A receptor shows a sharp border at the V1, V2 border and at the V1, V2 border here on the lower bank of the calcarine sulcus. And you see that the laminar pattern here is again different. Layer 3 to 4A are labeled and layer 4C. That's here in M2, layer 2 to 4A and 4C, and in alpha-2 receptor, layer 3 to 4A. And then we go to the glutamate receptors. The glutamate receptors also delineate the primary visual cortex, but in contrast to the muscarinic M2, to the alpha-2 and the GABA-A receptor, we have very low densities in the cortical layers where geniculocortical fibers are ending. Sorry. And more or less the same for the metabotropic glutamate receptor type 2-3, or for the nicotinic receptor, the alpha-4 beta-2 type, we can see the high labeling of a very, very small layer of the visual cortex, and this layer is layer 4C. So how do we know that this is primary visual cortex and the border? By comparison with cytoarchitecture. It is the easiest border to find in the human brain, the border between V1 and V2. It's a very, very clear border, and there is no discussion about this border. And you see that the receptors selectively label this border and have a special expression pattern in the primary visual cortex. I can tell you that this expression pattern is also found more or less in the same way in the primary auditory and primary somatosensory cortex. And when we go back to this section, then you see in the cortex a little spot here with high receptor density. And the question is this a primary cortex? And if yes, which one? It's the primary vestibular cortex. And it's also intensively labeled by muscarinic M2 receptors. The next question we had is this receptor pattern of the cortex in the general subdivisions of primary and other cortices, is this a conservative property during brain evolution, or is it a specificity of the human cerebral cortex? And so we compared a macaque brain and a human brain. And the time is over, or what? And the macaque brain was blown up. The macaque brain was blown up to the size of the human brain for easier comparison. Oh, oh. I have it. Yeah. No, I had it since without power supply since yesterday morning. Yeah, not too bad. Yeah. And now the European Canadian differences. Thank you. So what you can see here is, again, on the left side, the central sulcus and the Heschel gyres. I explained it before. And in the monkey brain, here is the central sulcus, and here is the comparable structure of the Heschel gyres. And you see that the muscarinic M2 receptor is also very, very dense in these. Yeah, it's okay. It's okay? Yeah. In these two regions. So we can state that it's okay. We can state that the expression of muscarinic receptors with very, very high density in primary sensory areas is a conserved property of the cerebral cortex, at least in primate brains. And we ask, is it in all primate brains? So in the upper row, you see here, you see the human occipital cortex, that of Macaca fascicularis, that of the vervet monkey, that of Calitrix, which is a New World monkey, and that of Galago, which is a very primitive or relatively primitive prosimian monkey in Africa and in Asia. And you can see that the primary visual cortex is heavily labeled in human brain, as I have shown before, in the monkey brain, the medial part and the lateral part of the primary visual cortex, in the vervet monkey. Now it's loading. Here. Yeah, it's also in the New World monkey here. And you can see it by a somewhat higher density in the Galago. So it's throughout all primate brains. And the same is for the somatosensory and the auditory cortex. You can see it at the same comparable sites in the brains of these monkeys. So receptors are very stable, very conserved markers of a special part of the cortex, the primary sensory cortex, and we can use such receptors for identifying cortices in comparative studies. But let's go back to the question of the big brain and the receptors. This is a section, a horizontal section through the big brain, and the colored parts of the cortex are different parts of the cortex, like auditory, somatosensory, but also higher cortices like V2, V3, V4, V5, and so on, or area 10 at the frontal pole. And our dream is that we can, and we are working on this presently, that we can, I will go back, yeah, just point to a part of the big brain, and then in the left upper corner, you will see the receptor distribution, for instance here, for glutamate receptors, and you will see a profile curve through the cortex. this is one step on which we already work. And we have now more than 44 different cortical areas which are characterized by their expression of 15 to 20 different receptor binding sites. 15 to 20. In most cases, we use 15 to 17 different receptor types. Why we use so many in the very same brain, I will explain it a bit later. Now, this is the SPART multi-receptor fingerprint. The concept was that a single neuron, a single layer, and a single cortical area does not only express one receptor type, it expresses many receptor types. And for the function of a microcircuit or of a cortical area, not only one receptor type is important, but the balance between as many different receptor types as we can measure and visualize. So what we do since some years is to map the receptors throughout the whole human brain and to map in each brain 15 to 17 different receptors. This, and the mapping is done at a resolution of approximately 20 micrometers isotropic. This is a very, very high resolution comparable to the resolution of the big brain, of the cytoarchitectonic big brain. And so we can fill up the big brain with functionally more important information, with the information about the distribution of the receptors, which are key molecules of signal processing. So each neuron has or expresses many receptors, glutamate receptors, GABA receptors, here, then different types with different function, then dopaminergic receptors, adrenergic receptors, and serotoninergic receptors. And therefore, we can measure in each cortical area the concentration of a specific receptor. And when we do it in a polar coordinate fashion, you see here the Ampa kinase NMDA receptors, which are all all glutamate receptors, the GABA A and GABA B, and the benzodiazepine binding sites, GABA receptors, M1, M2, M3, NACH, nicotinic receptor. These are cholinergic receptors for acetylcholine, alpha-1, alpha-2 for noradrenaline, 5-HT1A, 5-HT2 for serotonin, and D1 for dopamine. If we measure, register, and put all this data into a brain like the big brain or into the MNI reference space, then you can imagine that enormous amount of data must be handled, which is comparable to the data handled in the big brain. When we connect the different data points, then we get a figure which we call a multi-receptor fingerprint or a fingerprint. And the question is, are the fingerprints the same in the different brain regions or not? After having seen the figures before, you may predict that it's not the same. For instance, here in the caudate, that's the fingerprint of the caudate nucleus. That's the fingerprint of the putamen. Both fingerprints are very, very similar. Probably this is not. Yeah. Yeah, that's better. Yeah. The caudate and the putamen are two brain regions which have exactly the same cell types, the same connections, the same function. They are called, therefore, corpus striatum. And because these two parts of the brain, the caudate and the putamen, have the same function, they also have the same shape of the fingerprint. The area of the fingerprint is a bit smaller in the putamen than in the caudate nucleus. The reason for that is that the putamen contains more myelinated fiber tracts than the caudate. And myelinated fiber tracts don't have receptors. So the mean density of all receptors is proportionally lower. Therefore, the fingerprint is proportionally smaller than in the caudate, but it has the same shape. And now let's look to the motor cortex, area 4 of the human brain. You see immediately that this part of the brain has a completely different fingerprint. So the balance between the receptors is different from that in the corpus striatum. It means that all receptors are expressed in all 3 regions, but at different levels. And the level is something which I call the harmony of a cortical area. It's like an orchestra. If an orchestra is not tuned to the same harmony, then it will sound terribly. If it's tuned, then the different instruments speak more or less the same basic language. And therefore, I speculated that the functional systems in the brain which subserve the same function have similar fingerprints, and the functional systems which subserve different functions have different fingerprints. And let's see whether this is in fact the case. Before we come to that, I will make it quite short. We have to study the mechanisms behind the fingerprints. I said the fingerprint is an expression of a specific balance. And so we thought, let's challenge the normal balance by taking away one transmitter out of the sample of transmitter receptors we are looking for. And here we removed the serotonin 5-HT1A receptor by a conditional knockout model. Conditional knockout means that the knockout is done in the adult brain after having a long period of development of this receptor, which was quite normal. It says it loads. No, it doesn't load.
Sorry about this.
Yeah, now it loads.
It seems to be okay.
I'm sorry for that.
I'm sorry too. Seems to be okay.
Oh dear.
Oh, sorry.
So, in a conditional knockout, the 5-HT1A receptor gene is active during all the development during the young adult periods, and then we switch off the gene by tamoxifen injection. And we can see what a missing receptor does with all the other receptors of a fingerprint. And you see that some receptors are down-regulated compared to the wild type. For instance, the muscarinic M1 and the nicotinic alpha-4 beta-2 acetylcholine receptors, and other receptors are upregulated, like the serotonin 5-HT2, the GABA-A, and the noradrenaline alpha-1 receptor. So it's a very complex interaction between receptors in a receptor fingerprint, and there is a direct interaction between the expression levels of the different receptors. And I think because we cannot create such conditional knockouts for all receptors and study all possible interactions, I think this would be a very nice example for deep learning strategies to see after knockout of, let's say, 3 different receptor types, how the fingerprints are regulated, and to make predictions what could happen if a receptor type which we did not knock out in reality would be knocked out later, and how the different receptors are regulated. So, to come back to the question of functional systems and receptors, we studied, as an example, brain regions which are active during sentence comprehension tasks, a very demanding cognitive task. And all brain regions which are active during such a task are labeled in red, and in other colors there are brain regions which we used as internal controls, which have nothing to do with activation during sentence comprehension tasks. And when we do that and analyze the fingerprints and do a multivariate statistics, in this case a hierarchical cluster analysis of the receptors, you can see that all areas which are activated during a sentence comprehension task sit on one branch of the hierarchical tree. Other areas, multimodal association areas, are completely different, like these areas in the cingulate region in the prefrontal cortex and in the inferior temporal cortex, or in the inferior parietal cortex. And the primary sensory areas, V1, 3B, T1, which I have discussed before, they sit on a separate branch of the hierarchical tree. So receptor fingerprints apparently characterize neural systems with a similar functionality. And since the language function is on a dominant side more expressed than on the non-dominant hemispheric side, I have shown here the left hemisphere. Now let's go to the right hemisphere. If we look to the right hemisphere, then we see that the sensory areas, primary sensory areas, are stable and separated from all other cortical areas, that the motor cortex is separated, 4V, 4D, from all other cortical areas, and that the multimodal association areas are separated. Only the Broca region and the posterior part of the temporal cortex is left with similar fingerprints, showing that also in the right hemisphere, the Broca region and the posterior part of the temporal cortex is involved in language and has similar multi-receptor fingerprints. Now let's have a look to the visual cortex. In the visual cortex, we have recently identified two areas, FG1 and FG2, on the fusiform gyrus near to the early visual areas. FG1 has been later identified in collaboration with Karl Zilles and Kevin Weiner from Stanford as the word recognition area, and FG2 as the posterior face recognition area. And when you do multi-receptor fingerprints of these areas and of the primary visual, higher visual cortices, of the auditory cortices, somatosensory and motor cortices, you will see that the different modalities, auditory, visual, somatosensory, motor, is clearly separated by the fingerprints, and that the FG1, FG2 areas have similar fingerprints compared with the V3 and V4 areas, which are parts of the ventral visual stream, and face recognition and word recognition is clearly a function in the ventral visual stream. So also in this case, we could find a very good labeling. Now, back to the cortex and its layering. The question is, does receptors reflect cortical layers as defined in cytoarchitecture? On the upper left, we have a cytoarchitectonic preparation of the primary visual cortex with all the layers labeled, and myelin on the lower right. And we can just copy the cytoarchitectonic layers to the receptor distribution images of the very same area of the cortex, of V1. And what you see is that the exact borders of cytoarchitecture are not always reflected by the receptors. The receptor, each receptor presents you a different concept of cortical layering. Why is it so? Quite simple. The cytoarchitecture has as basis for subdivision and for recognition of cortical layers the stained cell bodies. And the cell bodies have only a minority of receptors on their membranes. Most of the receptors are found on the dendrites. And the dendrites are not in the same layer as the cell body. So the receptors give you an impression of the synaptic or dendritic structure of the cortex, and that is different from the cytoarchitecture. But it is functionally more important because there are the synapses. Here another example, the motor area, primary motor area, and if you compare, for instance, the muscarinic M2 receptor, second row, second from left, M2, then you see that there is a very low density, and the highest M2 receptor density is reached in the cytoarchitectonic layers 2 and upper 3. And when we go back to the primary, same place, second row, second from left, to the primary visual cortex, you will see that the receptors are expressed in completely different layers. And in the entorhinal cortex, we have a completely different cortical subdivision into layers. The reason is the entorhinal cortex is a phylogenetically old evolutionary old part of the cortex, which has a completely different layering structure. And it's not meaningful to apply the neocortical layering into 6 layers to parts like the entorhinal cortex, which has been done quite frequently in neuroimaging papers based on MRI scans. This is absolutely nonsense. The cortical layering of allocortical, the old areas, is completely different, and this is very nicely reflected here by the receptors. Then we can do measurements of profiles vertical to the surface for each of these areas. We can measure it in different brains. You see, or you hopefully can see, the standard deviation of these curves, because we have done it in several brains, not only in one. And the profile curves characterize in detail the laminar distribution of the receptors, which can be used for parcellations or for other questions in different areas. And then we thought, let's compare the receptor density over the cortical depth, over the complete depth from layer 1 to layer 6 with synaptic density. The synaptic density is labeled here as the straight black line, and the different receptors as the colored curves. And you will see that the GABA and glutamate receptor quite nicely follow the pattern of the synapses. But the M2 receptor has a different course. And so we can compare each receptor type related to the synaptic density. And we will find that some receptors clearly reflects the synaptic density throughout the cortex, like GABA-A and glutamate. Others do it also quite nicely, but not so clear, and others don't do it. The reason for that is that GABA-A and glutamate receptors are by far the most frequent receptors in the human cerebral cortex. So if receptors are at least partly associated with synapses as pre- or postsynaptic receptors, then they should clearly follow the synaptic density, whereas other receptors are modulatory receptors, and they can express or reflect the synaptic course more or less clear. If we study 44 different areas, and here on the left you see a receptor image. It's again the M2 receptor as a favorite example. On the right side, a section which you can also find in the big brain. It's a cytoarchitectonic section, and then you see that we can parcellate the cortex into different areas based on the receptor expression. So what should come up is a complete map of the human cerebral cortex based on each of the receptors or of all of the receptors in form of the receptor fingerprints. And here are fingerprints of many different cortical areas. And for the first time, we have subdivided the cortex into the supragranular layers, which are layers 1 to 3, including 3, the granular layer 4, and the infragranular layer 5 to 6. And we have done it because the supragranular layer are very often correlated with feed-forward connections, whereas the infragranular layer are correlated with feedback connections. And so we thought that we may also address this aspect of the cortex, the feed-forward and feedback aspect by receptor fingerprints. Not only the parcellation into different regions, the subdivision into primary or secondary or higher areas, but also the internal connectivity of the cortex. And if we analyze the fingerprints for supragranular, granular, and infragranular layers, for all 44 areas, you see that the fingerprints clearly separate these layers. So the layers have a completely different chemoarchitecture in that sense that they express more or less of the receptors in the different layers. They do not even overlap. They are completely separated, the 3 layers, with one notable exception. Under the 44 areas, Area 4 and Area 6. Area 4 is primary motor cortex. Area 6 is premotor cortex. So both clearly motor cortices have a completely different chemoarchitecture compared with the rest of the cortex. That's a fundamental, I think, a fundamental insight into the functional and chemical architecture of the cortex. And when we use the fingerprints of all 44 areas and we use the mean density of the receptor over all layers, we call it the regional fingerprint because we do not separate the layers, then we get a hierarchical cluster analysis like that. The interpretation will follow in a second. If we do it for the supragranular fingerprint, we can get another subdivision, and the subdivision is functionally meaningful. So we subdivide dorsal and ventral stream areas of the visual system. We subdivide auditory systems from somatosensory or from visual. We subdivide different multimodal systems and so on, and language areas. If we do that for the different layers, then we come to a principle of functional organization based on multimodal fingerprints. That is the following. If we have the neocortex, the complete neocortex, then the first functional system and the first functional cortical areas, which are very specific in their fingerprints and different from the rest of the cortex, are the visual areas, the early visual areas and the primary visual cortex, not the so-called highest visual areas like word recognition or face recognition. Then if we go through the hierarchical tree, then we have a subdivision of frontal, temporal, and parietal cortex on one hand, and of prefrontal, temporal, and parietal cortex on the other hand. And this can be subdivided, the latter one, into temporal association cortex and prefrontal association cortex. And if we go for the frontal, temporal, and parietal cortex and in further subdivisions, then we find that Broca's language region is different from higher visual areas, is different from parietal association cortex, from primary auditory and somatosensory cortex, and from higher auditory, higher somatosensory, and temporal parietal association cortices. You may ask, well, this is quite confusing. I think not, because the parallel subdivisions, the lumping together of the auditory and somatosensory cortex has a deep biological functional reason. basis. The basis is that both cortices are get mechanoreceptive signals in the inner ear and in the skin, whereas the visual cortices, the early visual cortices, they get the visual signals which are definitely not mechanoreceptive. So even this gives you a parallel insight into the cytoarchitecture, the chemoarchitecture, and the functional principles of subdivisions of the human cortex. Thank you for your attention.